thanks everyone for being here, for giving me the opportunity to give a talk. Um, the story I want to, and also if you're down here in the back, there is no microphone working, so you're gonna have to move in the front. So the story I'm gonna talk about today was part of my sort of last postdoc work, and it was done in collaboration with Carl Schoonover, who's now a PI at the Allen Institute in Seattle, and Andrew Fink, who's actually at Northwestern here, and that's part of the NITMB as well. Um, Great. So as Brent mentioned, I arrived in Chicago about 3 months ago, and well, actually, I should see this. I arrived in Chicago about 3 months ago, and one of the first thing I wanted to do was to try some deep dish pizza, because this was before I knew that it was, a tourist thing. And so really what I wanted to find out is which one of these two famous chain was the best. So I went to Malnati's, I went to Giordano's, and maybe, that's because I'm new or that's because I'm Italian, but, I really couldn't tell any difference between these two. So, but, people that have lived here for a long time, they start to notice the subtle differences. For example, the crust, how cooked is it, and the cheese, what kind of cheese they use. So you become an expert, and you form strong opinion. And indeed, even some news outlets, they feel like they should weigh in on which one is the best of this chain. So this phenomenon by which you, with experience, you become better at discriminating the subtle features of the sensory world is called perceptual learning in neuroscience. And it has been shown across a variety of different species and sensory modalities, including in olfaction in rodents, which is the relevant modalities for today's presentation. So we set out to find out whether experience with odors reorganizes the mouse olfactory system in a way that could increase olfactory accuracy. So here's the experiment. So we took mice in their home cage, and so we let them in their own cage free to sample a set of 4 neutral odors without any reward, hundreds of times per day for 2 weeks. And mice, even though there is no reward, they like to do this because I guess they're bored in their home cage. And so we call them the experience mice. And after this experience, we performed acute electrophysiological recordings using pixel probes in the primary olfactory cortex, which is also known as the piriform cortex. And in this experiment, we measured the responses to 2 sets of 4 odors each. This familiar set that the animal had experienced extensively, and another set of 4 odors, all of which were entirely novel and neutral to the animal. So to establish the discriminability of the responses, we performed population decoding from the responses of this brain region. And what we found is that the responses to the familiar odors were more easily discriminable compared to the responses of novel odors. So this is the decoding error of a linear classifier trained to discriminate the identity of the odor from the activity of the neurons in the brain region. And when we investigate further, what we found is that the reason for this decrease in errors is that these odors were higher dimensional. So again, I'm showing you the PTA dimensionality of this odor set, and was consistently higher for the familiar odors compared to the novel odors. But these odors were somewhat higher dimensional and decorrelated in their responses in the piriform cortex. So crucially, both of these effects were a result of this experience, because when we repeated these recordings in a new cohort of mice that did not have this same experience with the odors, then we didn't see any of these differences. Okay, so experience seems to make, yeah. We measure how many times the mouse goes and pokes these little nose parts. And the number range from like 1,000 to 5,000, depending on the animal, over 2 weeks. But I want to stress, for this naive animal, these 2 odor sets are both known. Okay, so experience makes this odor set, or the responses, I should say, this odor set, more discriminable and higher dimension. Okay, so this is in agreement with what has been observed in other brain regions, in the hippocampus, in other sensory cortices, in a variety of studies. Um, and so this seems to be sort of a general principle of the effect of experience in early sensory cortices. But the question is the piriform also circuitry, its connectivity reorganized in such a way that could support this um, effective experience? Because the piriform is a highly recurrent network, so it seemed reasonable to hypothesize that these changes were due to changes in the internal circuitry. Okay, so to answer this question, we needed a way to measure connectivity and responses in the same network. Now, measuring connectivity and responses is, as you might imagine, very, very hard. But we think that the piriform cortex presents an opportunity to solve this problem. Now, so for the next like 5 to 10 minutes, I'm gonna spend some time talking about the approach that we developed to detect connections from in vivo recordings. And so bear with me because that's then gonna be important to showing you what we found in this experiment. Okay, so the piriform cortex is a recurrent network 2 synapses away from the periphery, and these recurrent synapses are plastic in a slice. And for this reason, people have thought of the piriform as a learning center for olfaction. So to get into how we measure connectivity and activity, we sort of developed a new approach onto our old idea of inferring connections from the simultaneously recorded pairs of spike trains. Now, for those of you that have thought about this problem, this is very challenging and it has a fraught history. But you also might know that if the connectivity in this network is very sparse, then you might have better chances of this because you have less confounds. In particular, you have less chances of having common inputs, which is really the main confound that has hindered this approach over the past 50 years or so. So luckily for us, the piriform is a remarkably sparse network. 2 neurons in this network are estimated to be connected, even locally, only 0.1% of the time. And so to find these rare connections across the large number of simultaneously recorded pairs in our experiment, we developed a deep convolutional network that finds pairs of neurons that have a spike time cross-correlograms looking like this one. So let me explain you this one. So this term, spike time cross-correlogram, indicates the firing probability of a neuron relative to when the other one's found. So this is the latency of the spike of the pulse, let's say with respect of the pre. And in this particular example, you see the signature of a connected pair of neurons. So here you see a sharp peak that rises very sharply at around 1 to 2 millisecond latency. Okay, so to resolve this shape, we needed a lot of spikes, as you might imagine. So the recordings they're very long, between 6 and 8 hours. And then once you have this data, our trained deep convolutional network that has been trained to recognize this shape literally looks at all these cross-correlograms and finds the one that have this feature. And here are some examples. And whenever it does, I'm going to call this pair of neurons a putative monosynaptic connection. So this is a pretty strong claim to make because, of course, this is our extracellular recorded spikes. But here's why we feel comfortable making this claim. So first, we validated our approach using simulated network of leaky integrated and fired neurons. And I'm going to go a little bit in more details on this part. But overall, our approach detected connections with a very high precision, above 98%, and compared favorably to the state of the art. Our performance is this one in purple. Then we developed a set of analysis to estimate the error rate on our own recordings. So these analyses ask in one way or another, how likely are we to find a connection where there couldn't possibly be one? For example, here, how likely are we to find an excitatory connection outgoing from a likely inhibitory neurons? And altogether, these analyses indicate that our precision is, again, above 95%. And finally, we use a gold standard in vivo ground truth dataset collected by Dan English and Sam McKenzie with positively identified synaptic connections. And on this dataset, our approach did not get fooled a single time, although I should mention that this dataset only has around 30 connections to work with, so it's a very small dataset. Okay, so our approach is precise, meaning that if it says that the two neurons are connected, then it's likely the case. However, there is a trade-off in terms of it being also conservative. And to explain you what I mean, I'm gonna show you a little bit more details about the specific validation on synthetic data. So here's one benchmark that we did. So we take RNN, more precisely like a balanced network with leaking period and 5 neurons, sparse, and random connectivity driven by some external input. And if you simulate this network, you get this sort of asynchronous irregular activity. Now we can take this simulated spike and compute the spike time cross-correlograms. And here I'm showing you two instructive examples. So this one corresponds to an excitatory pair of neurons. You see this, again, this very sharp peak. Here, this bar is like 5 milliseconds. Here's an unconnected pair of neurons. And you see that they're also very correlated. And there's a peak which is also shifted to the right of 0. So if you do something naive, which is just looking for a peak to the right of 0, this would be a false positive because these two neurons are not connected. They just receive correlated input, which happen to be a little bit offset. So how do you get rid of this? Well, you can look at the fine features of this peak, and especially this sharp rise, which is really the signature of the synaptic connection. And if you do so, and that's what our deep convolutional network does, you get a much better precision and recall. So the precision here is a fraction of the identified connections, which are actually true connections. And the recall is how many of the total connections in the network you're actually finding. And as you can see, precision decreases as increase the recall, but our approach in purple does much better than a state-of-the-art method based on pure peak detection. If you can summarize this by saying, okay, let's say we want to be very precise and very confident that the connections are true. So we're going to put a threshold at 95% precision, so up here. How many of the connections you can find, and our method can find about half of the connections in this network compared to the previous method, only 15%. Yes? Yeah, so I mean, there's some sort of work that I'm not showing you, which is how we establish in sort of by physically realistic models that these are the features. But basically, the main features is that there's need to be a peak to the right of 0. So that's true for both of these. But the main features are, first of all, this peak is asymmetric in the case of a real connection. Second of all, this peak has a very sharp rise. And third, it cannot start rising before 0 or even at 0 because there's need to be a physiological synaptic delay. And you see here, this peak starts rising before, it's very slow, and it's very similar. Yeah.
Exactly, but because of the kind of very strong peak in beta sets, very modest in peak. But do you also see a very strong peak, we see that exit in the scale?
You mean the real connections or the?
In this and
Yeah. But do you mean like which one is a small?
Oh, this is a peak. I know peak. Who gets the big sets? There's some data very strong peak.
Yeah. You can find stronger peak than this. Yes, this I showed you because they are sort of similar and similar size. And so to show you that sometimes you cannot just look at the height of the peak. You're gonna miss too many connections. Yeah.
Sorry. What is?
is, let's say there are 1,000 connections in your network, what fraction of those you're finding correct? So if you put your threshold very low, you can find all of them, but then you're going to find a bunch of garbage too. Yeah.
Categorization of which kind of connections are less likely to take a.
Yeah, I mean, as you might expect, connections with weak connections are less likely to be picked up. That's the short answer. There is another factor, which is connections on two neurons that don't fire a lot, or from neurons that don't fire a lot, are also a bit less likely to be picked up. But that's something that is, both of these issues can be mitigated by having very long recordings. But still, there is going to be a bias towards strong connections from highly spiking neurons or two highly spiking.
This might be a question more for Andrew and Carl, but the sort of 6 to 8 hour long recorder for it, like how does that factor into your ability to identify connections, given that there's like unavoidable drip in the electrode, there's like, like what how are you thinking of this?
Well, I mean, the 6 to 8 hours long recording is crucial. I feel like if we cut the subsample the spikes to half, then we could add, I don't know, roughly half of the connection. It's not like linear like this, but more or less. So it's crucial to have long recordings, and that's why those guys are really working hard to develop chronic implantation of the probe that drifts as little as possible, so that you can have stable waveforms over time. Yeah, that's really the essential part. This was acute, granted, but it seemed like when they did the spike sorting, the probe seemed to have settled enough that there wasn't a very strong drift. But yeah.
And this number is what's the average fire rate?
The median is 2. The mean is like 3 or 4. Yeah.
Your negatives of this big mentioned mostly like centrically, or you also see your subject because in real world the EI is
Yeah. So in previous, you're right. In previous approaches, people detected mostly E to I. Here we detect roughly half and half. That's our estimate. It's hard to say because we don't have optotagging, but we can do some waveform-based analysis, and roughly half of the connections are E onto E, and the other half are E onto I. Sorry, I was talking about the piriform data. Yeah. Here we detect kind of, for some reason, in this network, E and I have the same firing rate, so we detect them equally. There is no bias. Yeah, and then I'm gonna move on after this. All right, I think my computer just. Okay. Is the firing probability, if you want. Yeah. So given that there is one spike of the presynaptic neuron, how likely it is that there's a this is 10 hertz as the CL bar. Sorry, let me go to this example, which was a little bit better. Yeah, here. So what you do practically, you align all the spike of the presynaptic neuron to 0, and then you count how many spikes in each pin you have of the post. Yeah. Okay, so this one quick other example. So this was like an unstructured random network. We also did this kind of validation for a structured network, where the connectivity induces strong correlations between neurons. In particular, we took what is known as a ring model. So where it's called because neurons are arranged the connectivity between neurons is determined based on the position of these units on this sort of abstract one-dimensional ring, in such a way that nearby neurons are more likely to have an excitatory connection, and neurons far away are more likely to have an inhibitory connection. And so this is the connectivity matrix, and crucially, this is sparse, meaning that even neurons that are nearby might not have a connection, right? So these two neurons are nearby, but still are not connected. Nevertheless, when you simulate this, this shows this bump of activity which drifts over time. Okay, so we can play the same game, and these are again two examples of connected pairs. One is connected and one is unconnected, and in particular, it's like nearby. So they have these strong correlations with some peak at 0, but no monosynaptic peak. And once again, our methods can detect connections relatively well, roughly 50% when we set the precision required at 95. And again, this is a benchmark that was built to be very challenging for this kind of connectivity approach. And the claim of this paper was that this was impossible to do. Okay, so to conclude this part, so this is an example data set based on a single recording. The black dots are the estimated position of the neurons, and here are the connections that we have inferred from this data set. There were around 600 connections out of about 1 million possible pairs. Now I'm going to spare you all the details, but we did the same thing for inhibitory connections. So here you're looking for downwards peak in the correlogram. So this is exactly the same plot as before. We did a new convolutional network, trained it, and validated the same way. And so in addition to the excitatory connection, we can also plot all the inhibitory connection here. Okay, so there's a lot more under the hood, and you can find all the details in our preprint. And if you have other questions about the connection inference, this would be a good time to stop, because then I'm going to go back to the experiment and show you how we use this method for the to analyze our data. I think there were already a lot of questions during the presentation, so seems like we're fine. Great. Okay, so again, for both the experienced mice, which had these 2 weeks of familiarization with the odors, and the naive mice, which did not have this experience, well, we performed these recordings and we used the spontaneous activity to infer the connections between the neurons. And then we use a separate phase of the same recording that was not used for the inference to assess odor responses to the familiar and to the novel odors. And now we are in the position to ask, what effect did this experience have on the network connectivity? So the first thing we did was to look for signatures of Hebbian plasticity, because there's been a lot of ideas in the piriform cortex about it implementing some sort of Hopfield network in which there would be sort of auto-associative learning and assemblies corresponding to different odors. So when we did so, we found this very nice, what it's called typically like-to-like connectivity. So what I mean is that the connection probability between two neurons increased with the correlations of their trial average responses. So here, for each pair of excitatory neurons, in this case, you can compute how correlated the responses are and compute how likely they are to be connected. And indeed, we find this trend overall. So this was very interesting for us because, first of all, it was reminiscent of what people have found in the visual cortex using completely different approaches. And also, it really looked a lot like an experience-dependent, wire-together kind of phenomenon. However, we also found the same result when computing the correlations across the novel orders, also the same upward trend. And in fact, we find it even across for the naive animal, which didn't experience any of these orders. So at least within this kind of passive experience that we considered, remember there was no reward or no reinforcement, like-to-like connectivity was not enhanced by this experience. And this is actually consistent with experimental results showing that actually you need some sort of reinforcement to trigger plasticity in the piriform cortex. This shows you the strength of the synapse, and you see that there's increased strength only for when mixture of odor was rewarded and not when it was familiar but unrewarded. So this is really consistent, and in some sense, this might be a good thing functionally, because excitatory Hebbian plasticity would tend to sort of correlate the responses. And remember, our motivation initially was to look for a mechanism that might underlie this decorrelation. So since we were interested in decorrelation, we thought to look at inhibitory neurons instead, because that's a classic way to decorrelate. So we took a close look at inhibitory neurons, and for this talk, I mean fast-spiking neurons identified by their waypoint. So here is one example inhibitory neuron, and for this neuron, we found one excitatory input and one inhibitory output. But when we look at this other neuron, we found many more inputs and outputs. And so we wonder whether there is some features of how these neurons respond to these familiar odors that can predict how connected they are. And we found one clear predictor. So let me show you the responses of these two neurons. So the one on the right, the one that makes a lot of connection, respond very differentially to these four familiar odors. So respond strong to one, but weak to another one. But the one on the left, the one that makes makes very few connections, it responds to all of them, but to all of them is sort of in the same manner. So this sort of phenomenon that I'm illustrating here, high selectivity versus low selectivity, holds across the population and relates to their connectivity. Here I'm showing you a selectivity index that measures how more like this versus more like that an inhibitory neuron is. And I'm plotting it against the connection probability in terms of both number of inputs and number of inhibitory outputs. And as you see, neurons that are more selective to the familiar odors are more connected compared to neurons which are less selective. And crucially, one more click, and crucially, this effect was experience dependent because we did not observe it in the naive hand. Yes.
It depends level, but like for this point connectivity, like parents can be impacted by how the fast-spike neurons are very high primary rate.
We look at that. So there is no correlation between this x-axis and the triangle grade of the neuron. Yeah, but that's a good question. That could have been a clear confound. Yeah. Okay, so we found this experience-dependent reorganization in which highly selective neurons are those that are also more connected. And now the natural question was, what came first? The connectivity or the selectivity? Meaning, was it the case that highly connected neurons somehow became more selective, or vice versa? Highly selective neurons that initially had few connections sort of developed new connections. And I don't know the answer. We don't know the answer to this question, and I'll come back to this at the end. But before, I'm gonna show you some modeling results, trying to study what effect this organization has on the behavior of an act. I think I have still 10 more minutes or so it should be fine in time.
Yeah, very good clarification. I think I missed it. This connectivity, is there directionality to it, or are you just finding it sort of like?
So in this case is the sum of the number of inputs and the number of outputs onto inhibitory neurons. But actually, the strongest effect is when you only plot the number of inputs. So it's interesting because actually the number of inputs and outputs is correlated. So we think that it is really mostly driven by the number of inputs. Yeah. Okay, so we wanted to study what effect does this organization have on the network behavior. So we built a sort of recurrent neural network model featuring an excitatory population and an inhibitory population of neurons, all two are connected with sparse connectivity and driven by external input. So this is a simplified model of the piriform. And now the challenge was that we wanted to have this relationship between selectivity and connectivity somehow built in this model. And this was actually more challenging than I thought it would be. And if one of you has some idea of how to do this in a principled way, I would be very interested. But because we couldn't find a principled way, we sort of used a machine learning approach. So what we did is we said, okay, let's consider all connections are initialized at random, but the intra-population connections are fixed, both for E and for I neurons. But the connection from excitatory to inhibitory and vice versa are trained. And so we designed a loss function that sort of aims at capturing this correlation. It is actually this correlation has to be close to 1, while also penalizing changes to the firing statistics of the network so that overall there is some level of homeostasis. And then we train these connections using gradient descent on this loss. And so let me show you what happens. So when you initialize the network, there is no relationship between in degree and selectivity index, which you would expect. And then as you train, as you do apply gradient descent, then you sort of start building this relationship. So there is nothing surprising here. That just means that our optimization works. But now what was interesting for us is that at the same time as this optimization was undergoing, and here I'm showing you the correlation between x-axis and y-axis, this correlation becomes the x-axis. Sorry, this is a bit tricky to plot. So as this correlation increases, we move rightwards in this x-axis over training. Also, the dimensionality of the familiar odors compared to the novel one increases. And notice that this was not plugged in the optimization at all. This is just a phenomenon that emerges just because you're introducing this organization in. And as you keep training, this keeps increasing. So now, at the end of this training, the model has the same organization or relationship between connectivity and selectivity that you observed in the data. And as a result, for free, we get this increase in dimensionality, which is also reflected in an increase in the coding performance or decrease in the coding error. And this is, just as a reminder, this was the result that we got in the data. So we sort of named this phenomenon, this is a work in progress name, but we think of naming it contrastive inhibition because it's reminiscent of what happens in contrastive learning, where in machine learning, where you pull apart stimuli that needs to be pulled apart in an unsupervised fashion. It is implemented by inhibition, and is also different from a simpler way of decorrelating responses, which is what you could do with global inhibition. And the reason we wanted to stress this difference is because we have good evidence that this is not what happens in the data. So let me explain a second. So one easy way to decorrelate responses is to overall crank up inhibition. So this pacifies the population activity of the excitatory neurons and increases dimensionality in general. But this is not what happens in the data. So in the data, the population sparseness or sparsity is the same for the novel odors and for the amino odors. So we have no indication that this is what's happening in this network. And this also, in our model that has this contrastive inhibition, we also have the same. Yes.
It is no acts all.
Yeah. No, I'm using sparse connectivity to initialize, and connections which are 0, they remain 0. So again, just to stress that this has a big correlation without a sparseness, which has some advantages, including the fact that it doesn't sort of affect the signal to noise ratio, which is the case when you sparsify, typically you decrease the signal to noise ratio. So we wanted to get a little bit deeper into the mechanism by which we decorrelated our sparsenate. And so to do this, we went back to our model and we sort of unwrapped it, meaning we only looked at the input from inhibition to excitation. And we realized that what happens is that now, because you have this wiring in which the inhibitory neurons that are selected to the familiar odors send more outputs, then if you're an excitatory neurons, you're more likely to receive inputs from those neurons that are selected with the familiar. So if you then compute the total inhibitory current, this is going to be more variable for this for familiar stimuli than it's going to be for the novel stimuli. So overall, you're going to have a larger variance of the inhibitory current onto excitatory neurons for the familiar odors. And indeed, in the model, we can check this intuition just by measuring the current, and this is the difference in variance, so much larger compared to short-full connectivity. So this is the difference in variance familiar minus novel. In the data, of course, we don't have access to the input currents, but we can at least try to estimate it. So we can take the responses and we can take the inferred connectivity and try to see what's the inferred variance. And indeed, we find that in the experimental data, if we infer the variance this way of the input currents, then we get the same effect for the experienced animal, but not for the naive. So this suggests that this is indeed the mechanism, this rewiring is indeed the mechanism linking the change in the connectivity to the change in the responses. Okay, so I'm almost done. Maybe 5 more minutes since I started 5 minutes late. This is more of sort of a perspective part. So just to summarize again, we found this experience-dependent relationship in which inhibitory neurons, which are selective to the familiar stimuli, become more connected. And we propose that this is a central component of perceptual learning, facilitating the downstream readout of the familiar stimuli that the animal has to sort of deal with in its recent past. But now I would like to go back to these two alternatives. So how did this happen in time? Something we can do and is to look at the model over training. Now, of course, granted, this is not biological learning. This is like gradient descent, but we can still get some inspiration. And so we can look at the beginning and at the end and the organization of the network. And here I'm showing you each dot is an inhibitory neuron at the beginning, and same dot, same neurons here at the end. You see that this relationship between selectivity and connectivity emerges. The arrows show the movement of sort of these neurons overtraining in this space. And what this shows, perhaps unsurprisingly, a posteriori, is that both things sort of happen. So here I'm highlighting three trajectories. So if each of these trajectory is the trajectory of an inhibitory neuron over learning, and you see that there are all cases are possible. This neuron here just was selected and just increased its migrate. Some neurons sort of change almost only their selectivity, and some neurons change both. So basically, all things are sort of possible, and you can try to. The only prediction you can really make are sort of this, that neurons that have a large selectivity at the beginning should increase their number of inputs, and neurons that have a low selectivity should decrease it. And vice versa, neurons that are highly connected should increase their selectivity, and neurons that are poorly connected should decrease. Yes.
Yeah. So, I mean, maybe if I squint, I tend to see sort of not monotonic trends that in degree goes down first before it goes up.
Yeah.
That's sort of not exactly the narrative you're selling me.
You're right. Yes. Here I'm only comparing.
But there's a mismatch she's ahead. But
yes, you're right. You're right. There could be a case. So if you only compare beginning and end, this is what you should see.
Yeah.
But you're right. If you really look at soft learning, you might also seeing non-monotonic trends. But at least once learning is saturates, then you should be able to compare beginning versus end. Yeah, true. Ultimately, to sort of verify these kind of predictions, you will need longitudinal recording, and that's what my collaborators are sort of working towards. So I'm going to show you just a teaser. These are just the very early days. But here's an example of one recording over more than 10 days with 1 hour of acquisition every day. The probe was chronically implanted. Here's a connected pair of neurons on day 0 of the recording. And in this example, the connection stuck around for a couple of weeks. And here's a different pair of neurons, same recording, in which there was no visible peak at day 0. And then suddenly we have an emergence of a connection. And also, there's a third connected pair where we have a connection on day 0 that seemed to decay over time. So ultimately, this is the kind of data that we need to resolve this sort of chicken and egg questions and to verify these predictions of our model. So with this, I would like to thank Karen Andrew and also in particular Shuzi Wang, who's a PhD student that has been collaborating with me on the sort of validation of this approach on synthetic data, and you for your attention. Yes.
In practice at decoration without publication.
Yeah.
But I seem to remember that Protector Iller et al. At Ace saw code. The funky coming out differences.
Yeah, that's a good point. That's true. And so when at the beginning I mentioned that decorrelation might be a general principle, sort of sensory cortices. In many of those cases, the story seems to be the simpler one, meaning that it's just crank up inhibition, and you might specify. This is not what we see here. I wonder it's because maybe the experience is more extensive. The animals are really in their home cage. They sampled this so much. Every time they're awake, they go in and sample the odors. So maybe it becomes, one could dare to say that it's less artificial, and you want to reach a steady state where now these odors are part of your ward, and you need to encode them with a high SNR. I don't know though. That's really pure speculation. Could be they're just different. This is a archicortex, not a neocortex. So
False.
Yeah.
Just like it's like false.
Yes, exactly. Yeah, that's. There was one single network realization. Yes. Yeah. It looks surprisingly consistent across realization, but yes. No. Oh yeah, probably. I mean, that's why I say I want to set a big disclaimer here. So this is not biological learning, not even a model of biological learning. This is just creating sense. Something that I want to do is look at what if there are biological plausible learning rules that could approximate this phenomenon and how to discover them. Yeah, that would be interesting.
Are there plans to do opto-tagging? I mean, it'll be great to know these work. Is there a reason why? Then maybe again a question for Carl and Andrew. What's the challenge for opto-tagging?
They really don't want to put in anything more than what they need to.
Yeah, it's just it's mouse brains are small. Don't jam too much in.
Yeah, I feel like they're very attached to the protocol for the probe insertion. That they don't want to mess it up with adding something else, the optic fiber in that case, I guess. I think I could convince them if it was very important, but let's see. I gotta pick my battles, you know. Yeah. Yeah, so there are some caveats. So you need to be in a regime where the selective neurons are selective to start. So if you the initialization of the network determines sort of the broadness of this distribution at the beginning. So if you're not careful, all inhibitory neurons might end up not being selective at all, and that is very hard to learn. So that's, for example, one failure mode that I encountered. So I need to sort of crank up the inhibitory to inhibitory connections so that you had some sort of competition as well. And so that made the inhibitory neurons a bit more selective. So that was, so there are some issues like that. The good thing is that the setup that sort of shows this robustly is statistics which are quite consistent with the one that we observed in the data. So we are pretty happy with that. But yes, it's not true. Ah. Yeah, maybe. I mean, I didn't think about it, but that could be interesting. Yeah, I didn't think about learning rule to set the initial condition, but yeah, that could be interesting.